Good afternoon, everyone. I'm Dr. Williams, introducing our speaker for Research Grand Rounds, and it really is a pleasure and an honor and a privilege to introduce to you Dr. Daniel J. Conklin. He's professor of medicine, he's a university scholar, and he's the director of our exposure and phenotyping core within the Department of Medicine's Division of Environmental Medicine. He comes to us after doing his undergraduate work at Wisconsin Whitewater and did a master's degree at Wisconsin La Crosse. And then he did his Ph.D. at Notre Dame in cardiovascular physiology. He has been a stalwart in terms of making the kinds of investigations that will really change the way we look at environmental medicine. He's going to talk today about 150 years of discovery of TRPA1 mediated cardiopulmonary responses to inhaled irritants. I'm so looking forward to it, just so I can figure out how to get around the smoke at the airport, which sets my lungs off. So I'm going to learn a lot today. Thank you so much for doing this, Dan.
Thank you, Dr. Williams, for the invitation and for the reminder. There is no free lunch, so but the hospitality is warm and real, and support for the collegiality and education of everyone here at the university, and that we're on the same team. So it's a pleasure to present to you. As Dr. Williams mentioned, I'll be calling TRPA1 TRPA1 just to make it a little bit easier than using the acronym. And yeah, I wear a lot of hats here. I've been here 21 years now, investigating essentially the area of environmental cardiology. And that is, how do inhaled materials influence cardiovascular health? But this will also extend to cardiopulmonary today, and you'll see why there's such an important connection between those organ systems. I want to acknowledge a lot of support. This is ongoing support, but we've been actively funded through COBRA grants and the American Heart Association Tobacco Regulation and Addiction Center for over a decade. And a couple of these studies will contribute to some of the work that you'll see today. I also want to acknowledge my exemplary staff. I work with 4 full-time staff personnel and 1 part-time UofL undergraduate. And we are housed in the Center for Cardiometabolic Science, which is in Delia Baxter. But we also have an inhalation facility in the MDR building on the 7th floor. So as way of an outline, I'm just going to cover kind of 4 parts to this talk today, some shorter than others. And there's no, hopefully it'll go smoothly enough. You don't know what hits you until it's over. But essentially it starts with this idea of, and I apologize for the mantle. I couldn't find women that have been involved in this specific pathway, but I know they were involved. But the story starts with Florian Kratschmer, and in particular, it'll come clear why I got so interested in him from a cardiovascular physiology standpoint, but also this cardiopulmonary relationship. He was an Austrian military physician and doing research in the 1870s. And that's when we started. And then we'll transition to 19th century. Eve Alarie was at University of Pittsburgh for many years, and then finish up with Sven Erik Jordt as we move along. So why am I so interested in Kratschmer? Well, he published a paper in 1870 that was in German, didn't get translated until 2001. So you've probably never heard of the Kratschmer reflex. You will soon, because as you can tell from these traces, Kratschmer recorded respiratory rate, heart rate, and blood pressure in both cats and rabbits. He did several of these using his hands to also monitor for changes. So he held the animals while he did some of these experiments. And part of the irritant stimulus that he used was the cigarette he was using. He would take a puff and then blow it in the face of the animal. So this was before Iacuk, of course. But you can't argue with his traces. Irritant stimulus onset is followed by apnea and complete cessation of respiratory rate, which returned over time. And similarly, heart rate was also decreased significantly and a corresponding increase in blood pressure. So I can't go into all the experiments he did, but this guy basically took apart these animals to figure out where was the sensor for this reflex. But the one experiment I will show you is he transected the vagi and found that the decrease in respiratory rate was not affected by onset or duration, but heart rate decreased. The bradycardia was totally lost, indicating that there was an autonomic control going to the heart, but blood pressure was not affected. So it's important to consider, one, he showed that the nasal mucosa was very important in this reflex and that the vagus controlled heart rate, but not respiratory rate. So it got me thinking about some of the work we've been doing over the years with electronic cigarettes and that I didn't know very much about the neural connection between what we breathe in and some of the reflex actions that may occur. And the trigeminal is infinitely fascinating. Besides the vagus, it is the most innervated or largest of the cranial nerves. And you can see here it innervates everything in the face and the head, including your auditory system, the nasal mucosa, of course, but also the dura mater and the tongue. So it is a major sensory player, but I was also intrigued by the naked sensory nerve endings that go into the nasal mucosa between cells, but also integrate sensory cell output. That was a fascinating aspect because these sensory nerves would contain receptors for some of the compounds that Kratschmer was blowing onto the animals. And in fact, you can monitor for responses from the epithelium by putting an electrode in there. And this was done by a colleague at University of Maryland, Baltimore County, where she was looking at things in e-cigarettes, including nicotine, menthol, and cinnamonaldehyde. And I just showed this to demonstrate that the nasal mucosa is far more innervated than the trachea in terms of responsiveness to a variety of compounds. And that got us thinking that these compounds or stimulation of the nasal mucosa is incredibly important to activation of the trigeminal and then systemic reflexes, including cardiopulmonary physiology. Now, Alarie wasn't really thinking about the mechanism behind the stimulus of these sensory neurons, but he was interested in utilizing this reflex to protect occupational workers who have to are exposed to a variety of irritants. And he basically created this concept of an odor safety factor whereby you could use detection thresholds in animals to predict what would be a valuable threshold limit value for humans. And he worked really hard over many decades developing this approach. And what I show you here is just one of his graphs of 25 different common inhalants that he studied over multiple doses. And on the y-axis, you can see that he was measuring the decrease in respiratory rate. And he primarily used rats, but this would work in mice too, as you'll see. And the x-axis was the concentration in ppm. So these are all gases, and you can see what happens. As you increase the concentration, you decrease the respiratory rate. So this is good evidence that the Kratschmer reflex is being activated, or at least the braking response is being activated. And using not a very complicated, RD50 conversion to the logarithm, and then putting in some safety to that, he predicted that the animal model could tell us what the protective level should be for humans. And so he looked at these threshold levels and he calculated his own. And you can see that he produced a very robust relationship between the decrease in respiratory rate and what should be a safety level for humans inhaling these materials. He didn't understand the mechanism behind it. He was not interested in that, but fast forward 4 decades and we're all very interested in how we sense our environment. And the 2021 Nobel Prize was given to 2 folks for their role in identifying the basis for some of our most important sensors, the TRPV1, and then the piezo touch somatosensation. Interestingly enough, both of these Nobel laureates worked on TRPA1 as well at the same time. And I'm telling people working with me now, we still have a chance to get a Nobel Prize for TRPA1. But it probably should have been included in this group, in part because the TRPA1 is one of a whole family of TRP receptors. It's the only one in its class, but you can see it has a very similar molecular structure as TRPV1. One of the important differences is this amino terminal presence of multiple free cysteines. And these are considered important for binding covalently agonists of TRPA1, and you don't find those in TRPV1. Of course, we know TRPV1 is the hot receptor or capsaicin pepper receptor. TRPA1 is the wasabi receptor or the horseradish mustard oil receptor. It's also known as the pain receptor. So if you get trauma, you cut yourself, that pain you feel is mediated through activation of TRPA1 in sensory neurons located throughout your body. And the other distinction between TRPA1 and TRPV1 is their permeability to cations. TRPA1 is a calcium primarily, whereas TRPV1 allows sodium and calcium to enter. But the amazing thing about TRPA1 is the chemical heterogeneity that determines its agonist. And this will also become important in the story as it continues to develop. But for those clinically involved, if you've ever used isoflurane, this activates TRPA1. It's very interesting if you think about it, that decrease in breathing response is something we see with some anesthetics, and this can be driving it through that TRPA1. But as I mentioned, the horseradish or mustard oil found in wasabi, cinnamon aldehyde is a strong agonist, formaldehyde and acrolein, 2 very abundant aldehydes that we've been studying for years, are both agonists of TRPA1. And of course, irritants to our airways. So I just want to touch on a little bit of data about acrolein and TRPA1 that we've worked on over the last decade based on some counteract grant. And this is a program through NIH to develop interventions post-exposure. And certainly very important in wildfires that there can be a lot of exposures, both occupationally, but also to the general public. And as we all know, the wildfires are going to continue. And acrolein reaches very high levels, both in cigarette smoke, but also wildfires. And firefighters have an incredible increase in risk for cardiovascular complications than any other activity. So we did a study several years ago that looked at lethality in mice relative to high level short-term exposure. And it certainly is lethal, and much more so in males than in females. And we found that interesting. We thought, well, why don't we look at whether or not the sex dependence is dependent on TRPA1? And we received these mice from Sven Jordt at his time in David Julius's lab. He created this transgenic knockout. And what you can appreciate is that males and females, there's no more protection when they lose TRPA1. And this becomes important because while we know there are mechanisms mediated by TRPA1, you can't live without it, especially under these conditions of high level exposure. So its main purpose is to provide protection against the cardiopulmonary system from exposures. So the decreased breathing rate is important for protecting your body from that exposure. But we wanted to ask a simple question: whether or not TRPA1 was involved in the Kratschmer reflex, as identified as a bradycardia and decreased breathing rate. But we also wanted to know whether or not endothelial dysfunction was involved with this activation. And the reason is pretty straightforward. This is an early biomarker of cardiovascular disease risk. It's been shown in cigarette smoke and secondhand smoke for decades. It's now been shown with electronic cigarettes. And we wanted to know what's the underlying mechanism and what constituents in these complex aerosols are driving this response. Here's just a schematic of our workflow that we typically do. We work with male and female mice. We perform exposures in our inhalation facility. We record data during exposures using radiotelemetry. We can collect urine for biomarkers of exposure. And then we do ex vivo assessments of vascular reactivity. Okay, if you didn't know it, electronic cigarettes make a very toxic aerosol. It is not water vapor by any stretch of the matter. It is a complex aerosol, lots of particles, lots of gases. And the basic generation of these gases comes from the fact that the e-cigarettes that contain eating fluids have propylene glycol and vegetable glycerin. Both of them thermally decompose to make these small aldehydes. And some of these I've already mentioned, formaldehyde and acrolein. And we've been part of the groups that have monitored for aldehyde formation under different conditions. And certainly no two e-cigarettes are alike, and they can generate vastly different amounts of these aldehydes. But you should be aware this is a toxic aerosol. But the first thing we wanted to answer then was, does e-cigarette aerosol trigger this braking reflex? And what you can see in this top graph is wild type mice in the open circles show a decrease in their respiratory rate almost to 50% after just a couple puffs, which is shown along the x-axis here in these little spikes. And then as soon as we stop the puff session, you can see they return back to normal. So this is a very rapid on-off reflex controlled response. But the filled circles are TRPA1 null mice. And they don't respond at all with a change in their breathing rate during this exposure. So that shows that the response to e-cigarette aerosol, even though it's an incredibly complex mixture, is absolutely TRPA1 dependent. So we looked at our two favorite aldehydes here, Trollian, also stimulated, and here it's a concentration dependent decrease in breathing rate, very robust, but rapid reversal. And similarly with formaldehyde, we see in wild type mice, you get that suppression of breathing rate and not in the null mice. So two aldehydes similarly depress breathing rate as that of e-cigarette aerosol. So we know then that these aldehydes are triggering this TRPA1 and that can lead to activation of the nervous system. We know the vagus is getting activated. I'm not going to show you the data, but we also show that bradycardia is stimulated by each of these agents as well. So we know the nervous system is getting activated, but we don't have direct evidence that the endothelium is becoming dysfunctional because of this activation. Well, at the time we were starting to conduct these experiments, a colleague at UCSF, no less, Matt Springer and his group showed that inhalation of e-cigarette aerosol, inhalation exposure to e-cigarette aerosol, marijuana smoke, conventional tobacco smoke, and aldehyde gases in rats would impair FMD, but not when the vagus nerves were cut. Again, repeating Kratschmer's experiment from 150 years ago to show that FMD impairment, for those that don't know, flow-mediated dilation. After 5 minutes of ischemia, you get a dilation you can measure non-invasively with ultrasound. Dr. Springer does this in rats, and basically he did not see the impairment. But he went a step further and he showed that a single gas, acrolein, would also produce FMD impairment. And while that was kind of a scoop on us, he hadn't shown that the connection between the nervous system and the exposure was TRPA1. So there was space for us to test our hypothesis. And at the same time, this study came out in humans showing that healthy young females who went to the cadaver lab had impaired FMD just 90 minutes after. And you can see here it's in A is the raw percentage change and then corrected for shear stress, which actually improved the relationship. So this, as far as I know, is the first study in humans where kind of attributed endothelial dysfunction to formaldehyde exposure. Now, we just published 2 weeks ago our work that says formaldehyde activates TRPA1 to contribute to e-cigarette induced endothelial dysfunction in mice. And so let me just show you the data. And this was work done in part by a summer research scholar program participant, Laura Fryer, in 2022. And she's now an M4. These are the wild type data. Again, isolation of the thoracic aorta, put that in an organ bath, and then you can pharmacologically stimulate for contraction with phenylephrine and relaxation with acetylcholine. And the acetylcholine relaxation is endothelial dependent, nitric oxide dependent in the aorta. So your control here is the air. The open circle shows a normal functioning blood vessel, but both PGVG, the e-cigarette, and formaldehyde significantly impaired that. And when we did a similar experiment in the TRPA1 null mice, we did not see that. The air actually was shown here. And in fact, the e-cigarette exposed aortas were actually enhanced in their vascular response. I'm not saying it's better. In fact, it's a little disconcerting to produce so much nitric oxide in the vascular wall. But we'll save that one for another day. we answered our question whether or not TRPA1 was involved and what aldehydes could stimulate endothelial dysfunction. So we have another long history of our work here at UofL and in the Envirome, where we're interested in particulate air pollution. So instead of the gases, we're interested in the particles. And we find particles, of course, in wildfire smoke as production from industry, vehicle exhaust. And in our inhalation facility, we can perform controlled particulate matter exposures and test whether or not TRPA1 is involved in changes in the cardiopulmonary system. So here's a schematic of our workflow. We also added in this model a hypertensive condition. Hypertension, as you well know, is the most common cardiovascular condition in the world. And certainly many epidemiological studies have shown that people who have hypertension and are exposed to air pollution do much worse. So we wanted to understand maybe there's a role for TRPA1 that is specific to the condition. What you see in the second panel here is our exposure. In this model, we go 30 days and we collect Louisville's particulate air pollution. We concentrate it 6 to 10 times, and the animals are exposed for 6 hours a day. And after their exposure each day or every other day, we're measuring non-invasive blood pressure. I just want to make a shout out to Dr. Lung-Chi Chen at NYU. He developed the system and donated some of this material to us 15 years ago when we opened our inhalation facility, and it is still working beautifully. I just talked to him this past weekend, and he was happy to see some of these data. Here last summer, the Canadian wildfires, if you remember them coming into town, we were running this exposure. It was nearing the end of our study, but these fill dots, you can see, go all the way up to over 1,700 micrograms per meter cubed in the chamber that the mouse was being exposed to. I think we reached several hundred outside during those days at our worst. Okay. Another medical student, Gabby Bonvian, is now in M3. She worked with us last summer to do the non-invasive blood pressure measurements with Lei Xiao Jin, who's been with me in the lab since 2016. And interestingly enough, if you look across these 4 panels, the only significant effects we got were in wild-type mice that were also hypertensive. And they actually got a more accelerated hypertension during cap exposure or the air pollution exposure. This effect was not present in TRPA1 null mice, indicating that the hypertension acceleration was being mediated by TRPA1. In this model, as you may well know, not only do they get hypertension, but within 30 days, you get significant cardiac hypertrophy. And that's demonstrated here that the amount of hypertension is associated very nicely with the amount of hypertrophy at the level of heart rate. Okay, and when we summarize these heart rate data for the end of this exposure, what you see is the Ang2 or the hypertensive mice definitely got larger hearts, but we couldn't distinguish an effective cap or an effective TRPA1 at the level of the whole heart. But what we did notice is in this cap group in the wild type that we got a more toxicological spread. That is, the variation was getting larger in that group, indicating that more was happening there. So we took the apex of these hearts from wild type mice and submitted them to the Sequencing Technology Center, and Dr. Melissa Smith worked through bulk RNA-seq, and then working with Dr. Richa Singhal in the Center for Cardiometabolic Science, we performed this pipeline analysis. And I know this is a busy slide, but there's only a few aspects of it that were particularly intriguing. And that is we saw no gene changes, significant gene changes between animals exposed, normotensive mice exposed to air versus CAP. No significant changes in genes, okay? However, in the hypertensive mice, and remember in the wild type mice where we saw accelerated hypertension in those mice with the air pollution exposure, we actually saw almost 1,000 genes change. 332 up, 664 down in that group. And it was more genes that changed than even hypertension alone in the air group versus the normotensive, shown right below it here. The other data then summarized here for these gene changes include this upset graph, which is kind of along the lines of the Venn diagram, but you can appreciate individual comparisons, how many genes changed just between hypertensive CAP and hypertensive air, only there, and then what genes were shared. So you can then parse out what was due to hypertension, what was due to CAP, and what are unique changes. And the volcano plot is also shown to identify the most extreme changes in genes that were occurring down on the left here and up on the right. And that's that 1,000 genes. So how do you break this kind of data down? One way is to go through KEGG and gene ontology pathways and form clusters. That is, put the number of genes that are related to pathways together. And so you see this functional enrichment through these cluster profiling. And what I want to point out here is that hypertension versus normotensive, you would expect to find large number of changes in muscle cell development, striated muscle cell differentiation, extracellular matrix, because the hearts are changing towards a more hypertrophic and fibrotic state. Okay, that's what we saw. Very convincing. How much of that carried over to the CAP exposure? Okay, and so all the black bars connect the ones that are shared in both groups. These are the top 20 clusters. The ones with stars are ones that uniquely occurred in that group alone, that exposure alone, and did not occur in the hypertension versus normotensive. I just want to point out 3 of them. In utero embryonic development genes, over 50 some genes were activated in that category. Covalent chromatin modification and histone modification. So we're going to chase down these genes because this suggests epigenetic remodeling was occurring under those conditions, and whether or not those were related to TRPA1 remains an important question to address. Okay, this past summer, following up on that to look more closely at changes in the heart was done by another summer research scholar program, M2 Matthew Jennard, and he basically quantified left ventricular area rather than the whole heart weight. We wanted to look what was happening in the left ventricle because our gene changes came from the left ventricle apex. He also looked at interstitial and perivascular fibrosis. And I think what you can appreciate is that the Ang2 CAP group was the top in every category. So we definitely were seeing enhanced hypertrophy and enhanced fibrosis going on in the hearts of those mice exposed to CAP. And further work will go on connecting the gene changes with these structural changes. We've been accepted to the American Heart Association to present a poster in Chicago this fall. And now Act 3 lest you think everything we talk about is bad for the cardiopulmonary system, we've been interested in studies of greenness and where you live determining your cardiovascular health. And certainly the Green Heart Project that Aruni Bhatnagar has been heading up is essentially to test this idea that there are things in the environment that are also good for us. And that if we can balance these things out or understand better how they're good for us, we can push that scientifically. And what I show here is a workflow that we did on a project to investigate one of the most common compounds released by coniferous trees. Alpha pinene gives that nice pine smell. And when you forest bathe, you recognize you're in a pine forest through this alpha pinene. It makes up almost 40% of the hydrocarbons that are released from conifers. So it's highly abundant. But we asked the question whether or not we could detect its metabolites in the urine, because we wanted to also develop a biomarker of greenness exposure. Okay, and you can use mice because they're not exposed to alpha pinene through any other pathway, okay, than our inhalation. And what we discovered were a couple different phase 1 alcohols, and I'll go into these in a little bit. We then tested in isolated blood vessels of naive mice for beneficial relaxation. And we examined the mechanisms for that. Okay, here's just a workflow of our analysis for HPLC and mass spec, and then isolated blood vessel, and the protocol pre-contraction with phenylephrine in aortas and superior mesenteric artery. They have some things in common, but not all things in common in terms of relaxations. And what we found in the superior mesenteric artery, what I'm showing you here are data from a control mouse, a mouse vessel that's treated with L-name. This is an inhibitor of nitric oxide synthase, causes an increase in tension, and then eNOS knockout mice, because we wanted to know whether or not this was working through activation of an endothelial specific pathway. Okay, and I won't show you all the data, but what you can appreciate in the summary graph down below is that in both cases where we knocked off eNOS, either pharmacologically or genetically, we did not get much of a relaxation with alpha pinene, but the control gave us a very robust, almost 70% relaxation, up to 300 micromolar. When we do the aorta, we don't see relaxation until 300 micromolar. Okay, so it's not sensitive and it's not dependent on eNOS. So we've kind of looking away from the aorta and focusing more on the superior mesenteric artery. And we didn't really have a hypothesis for this experiment that the metabolites would stimulate anything in the vasculature, except for the fact that when you take in these compounds, they're almost always metabolized very rapidly. And this is the first pass metabolism would go to a phase 1 alcohol. And what we were surprised to see was that actually erythritol and verbinol, both alcohols, produced concentration-dependent relaxations that were dependent on the endothelium and dependent on nitric oxide. Okay, so there's myrtenol and verbinol. Totally shocked us that we were getting responses. Most of our professional lives have suggested that metabolism removes the reactivity of compounds. So this intrigued us greatly. We wanted to know, how are these things activating the endothelium? So we looked whether or not TRPA1 was involved. And so we used, again, wild type mice and TRPA1 null mice. And you can appreciate that the wild type mice, nice relaxation with alpha pinene, significantly impaired in the TRPA1 null mice, indicating that TRPA1 on the endothelium was being activated by alpha pinene. Verbenol was somewhere in between. Mertenol showed no dependence on TRPA1, indicating that the metabolites may be responsible for relaxations in part through TRPA1 and through a novel mechanism. Just to let you know, TRPA1 is found in lots of different cardiovascular cells, not just in sensory neurons. We get very good staining for TRPA1 in the endothelium of the superior mesenteric artery. Here it's localized to isolectin, which is another marker of the endothelium. It's not found in the aortic endothelium, which explained our results why we didn't get relaxation with alpha pinene there. This opened up a whole set of questions about how these compounds might be stimulating relaxation. It's very difficult to do every possible compound through the isolation of blood vessels. That's a very slow way to walk through all the potential actors. So we did a project this summer, again with Jake Baldoff, again a summer research scholar program participant, and he worked with a colleague, Zhengji Che and Rob Monson in the Cancer Center to look at in silico ways to test how these compounds may be activating TRPA1 or any other target. So this is a workflow that the team developed so that Jake could start to screen for places that these compounds may bind to TRPA1. And what I'm showing in the middle is the model structure of TRPA1 as a channel, and the terminal or N-terminal cysteines are shown over here. 621 is a known binding site for that horseradish mustard oil compound, AITC. So he compared some of the metabolites that would bind there. And then there's also a transmembrane 4, transmembrane 5 binding pocket that has been shown to activate TRPA1 through a non-covalent mechanism. And this is the positive control there. So we started with that, showed he could model that binding, and then looked at a number of different metabolites. And to make a long story short, Jake did a great job taking metabolites of limonene, another very common biogenic VOC, as well as alpha pinene, and looking at their binding scores at these two different sites. This is the cysteine site, C621, and then their predicted ability to stimulate endothelial activation. And a long process to go through it, but as you go down in binding score, that's better. And as you go up in the log of the EC50, the better. So the compounds over here would be predicted to be better activators through this one site than those on the other side. And what you can see he has in these kind of orange triangles, alpha pinene here, limonene here. So alpha pinene is not terrible in terms of its binding, or its binding is not great, but its activation is good, its potency is good. Verbenol and Mertenol are way over here. So not great binding and not all that active, but they're still activating. Now this is one site, the cysteine. So that's the covalent modified one. But nonetheless, he got a lot of hits from a lot of metabolites that I don't have time to go through. But he also modeled the other site, the transmembrane that is due to non-covalent interactions. The beauty of this is he was able to do all of this testing, and now we know what are some really good targets to go and test in the blood vessels to see, do we really get an endothelial response without doing these hundreds of proposed metabolites. So that was a highly productive summer project with Jake. And let me just summarize here. I've tried to show that the irritants from a variety of sources activate TRPA1 dependent Kratschmer reflex, which has then not only pulmonary but cardiac responses, including endothelial dysfunction. And our research indicates it may be involved in lots of other outcomes as well. Certainly migraine or SIDS could be issues to address through this pathway. The TRPA1 dependent effects are complicated. They can be mediated locally because TRPA1 is on lots of different cell types, not just sensory neurons, or systemically via the nervous system and or circulating factors. We've used only whole body knockout mice. There's a real need to target cell-specific TRPA1 deletion so that we can figure out which site is responsible for what outcome. And then I think we really opened up a good avenue here with in silico approaches to target and develop greenness biomarkers of health. And I'm happy to answer any questions.
That was remarkable, Dan. Your first question is not a question, it's a comment. Really impressive that you include these students. It's changing their lives completely to have successful research that is publishable and you start them on a career of investigation. So thank you so much for doing that. And I was wondering if there are other people who could contact you to figure out how to incorporate those students in their lab. I'm sure there's a program for the summer research and there's an application process or something, let people know, or you.
So I've been doing the summer research scholar program. I'm also an MPI on the T35, which has a subset that are interested in environmental health sciences. These I had 3 students this summer, and none of them were interested in the distinction in research track that is offered in medical school here, despite being very, very good and committed to their individual projects. I couldn't even get them to commit to that. In the 18 years that I've had students, I've had one come back after their first summer in their 4th year for an elective for 1 semester, and I've had students continuously every year. That's okay. I don't, some of them became interested in cardiology through this and dedicated their lives to that. The one that did come back, she became a cardiologist at Cincinnati after she completed here. So I was, certainly supportive of that. I think it's important to get exposed to science. Most of the students that do the summer, they just don't want to do nothing, right? They're very driven. They want to have an experience. So I think this may be the last time they interact with science and scientific thinking, but we try to promote that and plant that seed. But the research distinction in research track, we talk about it. I know they talk about it during orientation with them, and then they bring it back up related to the summer research scholar programs. But I was surprised this year that nobody was interested in. Laura Fryer, 2 years ago, she definitely was on distinction in research. She was just on the paper we published with the formaldehyde, and she's still very interested but it's busy.
Right. Well, thank you for doing it. And if we can send a link around to get people to sign up, for their labs or their clinical research projects, it'd be great. I do have one, more academic question for you about this process. Interested in the Kratschmer reflex and Alarie's work, but looking at those graphs, there's somehow an assumption, unlike the pollutants resulting in LVH, where you can actually track it. There's somehow an assumption that the cardiopulmonary response, the drop in heart rate or blood pressure, if that occurred, was somehow going to predict what the molecule would do. I mean, we're talking wood alcohol versus grain alcohol. So the grain alcohol will make you drunk. It'll bother your liver if you keep doing it.
Yeah.
The wood alcohol will get you drunk, but it makes you blind. And
Yeah.
so there's your respiratory response. I'm just not getting it why you would feel like somehow doing all those chemicals was going to predict what would happen outside of it. I mean, because it's not the target. The cardiopulmonary may not be the target.
Yeah, and I apologize if I didn't make it clear. He was solely interested in inhalation of these compounds.
Right.
So if you ingest them
Yes.
or give them dermally, totally all bets are off. He was solely focused on using the animal respiratory response
Yes.
to codify that the assay could be useful for occupational health
I see.
and protecting people from getting exposed. I'm taking it to the right at a safe level. And so he thinks there's a very tight connection between how much you breathe in and this response. Because if you're triggering this response, it's clearly too high. Not that the response itself will cause the injury, but that it's indicating this is a level that we should not be encountering.
I'm sorry, I think I left that part out, but that's exactly what I'm saying is that he was assuming that the inhalation response was somehow related to the toxicity so that you would set a safe level.
Yeah.
But I mean, for carbon monoxide, it won't work at all. And
Right. Well, these are occupational issues related to that inhalation.
Right.
Now, if you do something outside of work that is more hazardous, I mean, you can't actually predict that.
Right. So yeah.
Yeah. So I think that is a more limited thing, but I wanted to expand on it to the sense that this is a relatively under-described reflex that has very significant systemic complications associated with it. And certainly in e-cigarettes where people are using them with abandon, it is like we need to sound the alarm.
Fascinating area. Other questions? Yes, sir.
It was great talk. I have a few questions. This mice, track one knockout. In steady state condition, do they have any phenotype? Because you challenge them with different irritants. It is the first one. Question. Second, you show very nice data on expression of these receptors on endothelium.
Yeah.
It is also expressed on endothelial progenitors. And maybe another question, because there is a very close link between endothelium and hematopoiesis from hemangioblasts. Are these receptors expressed also in hematopoietic cells? Did you look, for expression in different tissues, including marrow?
No, that's fascinating. Now it's certainly not on all endothelium.
Yeah.
But it's localized to different vascular beds.
Yeah.
I don't think that precludes that it could be on progenitor cells, but for some reason in those places, it doesn't express in the adult endothelium in some vascular beds, but it does in others. But I can't answer that question. We never looked. We could easily do that in flow cytometry to look for TRPA1 on the circulating progenitors. It was a good thought. Thanks.
As you shown, and we also know that high circulation of angiotensin II causes cardiac dysfunction, hypertrophy. But at the same time, angiotensin is known to cause targeting several organs such as the skeletal muscle, kidney, through regulation of inflammatory cytokine. have you looked at all those tissue and what is the role of inflammatory cytokine in regulation of, exposure to your cap?
Yeah, I mean, there's certainly a lot of organs we can look at. It's a pretty dramatic hypertrophic and fibrotic response we see with angiotensin II.
Yeah.
It didn't seem to be increase the dependence on TRPA1 other than that kind of acute hypertension we would see after exposures, which ultimately didn't turn into major remodeling aspects more than just Ang2 itself. So to the degree that we're focused on how much of the mediation is through TRPA1, probably not going to look into every organ that Ang2 is affecting because it's all of them. Yeah, there's no question.
Another question, in our recent study, we showed that angiotensin II high circulation not only affect left ventricle, but also the right ventricle, which leads to damage to the fibrosis in lung. What is your opinion about it, and have you done it?
Well, I didn't show the data, but the lungs definitely increase in mass.
Receptor?
No, they increase in actual lung weight to be a length in the Ang2 model. So there's definitely no question evidence that Ang2 is not only causing left heart, but right heart and probably pulmonary congestion and injury there. So no question, it influences every organ. We have more specific data on trachea, aorta, functional data. Again, Ang2 does something different than CAP does, actually. Together, they look like they're additive, but in both cases, not TRPA1 dependent. So that's about, but I would say that Ang2 has its own remodeling pathway. It looks like CAP does something very different. But remember when we did CAP exposure alone in normotensive mice, no gene changes. But when we combined it with hypertension, they went crazy. And the clusters suggested probably epigenetic remodeling, which was not something that was happening just with Ang2 by itself. So we think that's a very exciting area to follow up on.
Yeah. Right.
Yeah.
Kind of related question. So the hypertension that you showed in your slide, was it mostly driven by the muscle hypertrophy that you also showed in your slides, or was there some other factors?
Well, we only measured non-invasive blood pressure by tail cuff, but the assumption is, it was driven as diastolic increases of around 20 millimeters of mercury. So I would infer that that's systemic resistance is the primary reason they get hypertension. Yeah.
Oh,
Please.
so correct me if I'm wrong, but cysteine 621 is on the intracellular side of the transmembrane channel. And so it makes sense that metabolites of your exposed irritants that are electrophilic might interact with the nucleophilic cysteines. But when when they're inhaling the electrophiles, how do you envision it getting in and targeting these intracellular sites?
It's a conundrum. Certainly the level of compound that you have to activate through the covalent mechanism has to get in. You consider these are small molecules, many of them. They actually could probably fit through some channels. Certainly they diffuse across these membranes. So we've measured many times intracellular proteins adducted with acrolein after exposures. So there's no question it gets in. So when these are activated by covalent modification, sets off the opening of the channel, calcium comes in, and then depending on the cell type, you get an appropriate response to that activation. But it seems like not the best engineering for adducting an extracellular compound. You would know better if you had that on the outside, but evolution is not about bioengineering. It's about what works in the context of where organisms find themselves. The more interesting thing is this role of the non-covalent binding these folds. They seem to be almost as robust as the covalent modification, not as potent, but robust at opening. So we think that's a very exciting avenue. But I agree with you, the metabolism part would actually make that electrophiles less likely if they get glutathiolated, for instance, with interacting with the free cysteine. But nonetheless, it's been established that those are involved when they're adducted with opening the channel. Yeah.
Wonderful. Thank you so much.
Thank you.
Thanks, everyone.